Hi everyone. In this video, we will discuss multi-particulate drug delivery system. Multi-particulate such as pellets, beads, and granules are a key part of modern drug delivery. They offer major advantages: flexible dosing, controlled or modified release, better distribution in GI tract, reduced risk for dose dumping. However, multiparticulate behave very differently from tablets, both in formulation design and dissolution testing. Each unit releases drug independently, so their hydrodynamics, floating behavior, and release patterns are more complex. That's why multiparticulate require a different testing mindset. These units include microgranules, coated beads, non-permeable seeds, and MUPS, multi-unit pellet systems. Typically, particle size ranges from 200 to 2000 micron. They are usually filled into capsule or sachet or compressed into MUPS tablets. Why are they better? Because drugs release happen from hundreds of tiny units. They provide smoother and more uniform release, lower risk of dose dumping, better distribution throughout the GI tract, and depending on coating and design, they can deliver IR, DR, or ER type dose. Let's define the main type of multi-particulate system. Each type has its own release mechanism and coating strategy. Pellets. These are spherical particles produced by extrusion spheronization. They are often coated for IR, DR, or ER release. Beads. Here the drugs is layered onto sugar or starch cores. Release is controlled mainly by polymer coating. Mini tablets. These are very small compressed tablets filled into capsules. Each unit behaves like a mini dosage form. Sprinkle capsules. These capsules can be opened and sprinkled on food or liquids, useful for pediatric and geriatric patients. Multi-particulate systems offer major clinical benefits. Because the dose is spread across many small units, they provide reduced variability in drug absorption, lower risk of dose dumping, and more uniform GI distribution, leading to a smoother plasma level. They are especially useful for patients with dysphagia, pediatric and geriatric population due to flexible dosing. One powerful advantage is the ability to combine multiple release profile in a single capsule. For example, IR beads for fast onset, ER beads for prolonged action, and even if few units fail, the system remains robust because hundreds of other units continue releasing the drug. Polymer selection is the key driver of release behavior in multiparticulates. The type of polymer determines the function of the system. HPMC or ethyl cellulose for extended or sustained release, Udrazit L or S for enteric protection, special polymers for taste masking. How these systems are made: drug layering onto inert seeds for beads, extrusion spheronization to produce pellets, followed by polymer coating to control release. This coating defines whether the product is immediate release, delayed release, or sustained release. Finally, different coating fractions can mixed and filled onto capsule. For example, combining IR and ER pellets in one dosage form, the polymer controls the release. The process builds the system. Release from multi-particulate is finally controlled by coating design. Key factors include coating thickness. Thicker film slows release. Plasticizer content affects film flexibility and permeability. Pore formers create channels for drug diffusion. Swelling polymers hydrate and regulate release rate. Common release mechanism: diffusion through the polymer membrane, osmotic pressure pushing drug out of coated beads, polymer hydration and swelling, controlling drug escape, pH-dependent dissolution for enteric coatings. In most products, multiple coating layers are used, each serving a different purpose: protection, delay, sustained release, test masking. So remember, multi-particulate rarely rely on a single mechanism. Combination systems are the norm. That's what makes the powerful, flexible, and clinical effective. Multi-particulate system can behave unpredictably in USB apparatus too. Because of their small size, they are very sensitive to local flow variations inside the vessel. Common challenges include floating beads or pellets due to low density, coning at the bottom of the vessel, leading to poor exposure to shear forces, agglomeration or sticking of particles, inconsistent mixing because the tiny units follow microcurrents, filter clogging caused by polymers, fragments from coatings. All these effects can cause slow or incomplete release, high variability, or poor reproducibility. Choosing the right apparatus is critical for multi-particulate dissolution. USP apparatus 2. This is most commonly used system, but small pellets may suffer from floating or coning if they properly not controlled. USB apparatus 1 basket helps prevent floating, especially for low density beads. However, it may restrict natural movement of pellets. USB apparatus 4 flow through cell. This provides uniform media exposure and it's excellent for extended release beads, low solubility API, complex polymer coated pellets. USB 4 often gives better discrimination and reproducibility for multi-particulates. Sinkers use them only for large multi-particulates. Small pellets should remain free-flowing for realistic hydrodynamics. Match the apparatus to pellets behavior for meaningful results. Enteric and extended release pellets require different testing strategies. For enteric pellets, there should be no drug release in acid. That's why we use pH stage testing, pH 1.2 to pH 6.8. This confirms acid resistance and proper intestinal release. For extended release pellets, long duration testing is critical, often 8 to 24 hours. You must maintain sink conditions throughout the test. Always confirm drug stability in the medium. Important cautions: avoid excessive surfactants, they can mask coating defects. Use biorelevant media when needed for physiologically meaningful results. The goal is simple: confirms the coating performs exactly as designed. No early release, no dose dumping, and controlled delivery. Sampling strategy is critical for multi-particulate dissolution. Pellets can settle unevenly, so always sample from a consistent location. Ensure uniform dispersion before each withdrawal. Typical sampling points for IR beads: 5, 10, and 15 minutes to capture rapid release. For ER beads: 1, 2, 4, 6, 12 hours for extended as needed based on release duration. Filtration is equally important. Perform filter winding studies, use proper pore size, ensure pellets do not enter the syringe or filter. This prevents false high or low results, clogging, poor reproducibility. Good sampling plus correct filter equal to reliable multi-particulate dissolution data. From a regulatory perspective, USP and FDA expect dilution to be robust, no matter how the product is used. For sprinkle capsules, Beads are designed to be sprinkled on soft foods like applesauce or yogurt, swallowed without chewing. That means the coating must be strong enough to resist crushing and chewing, withstand moisture from food, survive handling during administration. Regulatory expectations include consistent dissolution performance, no damage to coating during sprinkling, no premature drug release. In some cases, dissolution testing may be needed, food effect simulation, or modified media to reflect real use conditions. Key risk to control: bead damage during handling can destroy the release profile. The coating must protect the drug until it reaches to the target site. Discriminatory dissolution methods are critical to SUPAC MR and ANDA submissions. Regulators expect your method to detect changes that can impact drug performance, not just pass good batches. To prove this, we intentionally test challenged batches such as undercoated versus overcoated beads. Batch with different polymer concentrations. Pellets made under different spheronization conditions. A scientifically designed method must clearly separate these variants, so slower or faster release where expected. Detect real manufacturing changes. If your method cannot see these differences, it is not truly discriminatory. A good dissolution method protects patients and satisfies regulators. To close, multiparticulate follow the same core principle as IR, DR, and ER formulations, but with greater flexibility and safety. Systems like pellets, beads, and sprinkle capsule improve dose uniformity, reduce dose dumping risk, support pediatric and geriatric use. Remember, drug release depends mainly on coating design. Common challenges include floating, coning, and filtration issue. Your apparatus, media, and sampling strategy must always match the release mechanism. When tested correctly, multiparticulate deliver reliable, predictable, and patient-friendly therapy. Modern formulations are moving towards multiparticulates, and so should our dissolution strategies. Thank you for watching this video. If you found this explanation helpful, please like, share and subscribe, and stay tuned because in this upcoming 62 video series we will explore through dissolution method development, validation, troubleshooting and many more real industry example. In next video we will discuss orally disintegrating tablets, special dissolution challenges. Post your question in comment section. See you in next video.